Hello, everyone, and welcome to phyloseminar.org. The current theme is microbial genetics, and this is the first talk in a series of 3 talks on that topic. Please use the YouTube live comment box to ask questions. Today's speaker is Artem Babaian. Artem's PhD is in medical genetics with Dixie Major at the University of British Columbia, focused on the role of endogenous retroviruses in the development of human cancer. At the onset of the COVID-19 pandemic, as an independent research scientist, which I think means unemployed, he initiated the Serratus project with the aim of reanalyzing all the world's RNA sequencing data to uncover the origins of the SARS-CoV-2 and all novel RNA virus species. After starting for a brief few months at the University of Cambridge as a Banting postdoctoral fellow, he joined the Department of Molecular Genetics at the University of Toronto as an assistant professor, where his new lab focuses on characterizing the complete biodiversity of the Earth's RNA viruses. Welcome, Artem, and thank you for participating.
Thank you very much. Hello, everyone. My name's, I guess as Eric introduced me, I'm Artem Babaian. And the laboratory in the University of Toronto that I've started is called the Laboratory of RNA-Based Life Forms. And it's my pleasure to share with you some of the research that we're doing. And so the title is humbly called Entering the Platinum Age of Virus Discovery. Oh, let me get that. So I usually like to start, especially when I'm talking to virologists, with this kind of framing of the type of work that we're doing. So computational virology, I view this as the vanguard of modern virus discovery efforts, right? So what we do is going to be a little bit different than what people in like molecular or wet virology do. And there's definite strengths to computational virology, but there are also limitations, and we're going to be running into some of those limitations in the work. And really, I think it's kind of balancing these two fields. And so, if you look historically, virology, since the discovery of tobacco mosaic virus, has really been flourishing since, the 19th century. And then with the advent of PCR and molecular techniques in the 70s, molecular virology really took off and has been essentially dominating the field. And computational virology has been like the little cousin of the kind of big virologies. But I think that this is, that may have been where we were, but in the future, I think computational virology is going to rapidly accelerate the amount of data and the amount of insights that we can gather. And the real crux of computational virology is taking those like key nuggets of information and folding them back so that we can get really good molecular insights, and so we can advance clinical and cell virology as well. So with that, I will introduce you to the Serratus project. So like many COVID-19 projects, this began at the beginning of the pandemic. And what was obvious back then, so kind of put your frame of reference back to like February, early March of 2020, was that this thing was going to be a society shifting event, COVID-19, of course. It was clear that, this was an all hands on deck type of endeavor. People were spending billions of dollars, and we could see for the first time how this virus was evolving in functionally real time and unfolding across the earth, right? So billions of dollars were spent on genomic surveillance. And I had a bit of a different question, not so much where this virus was going, how it was unfolding, but where did this virus come from? What are the evolutionary and proximal origins of SARS-CoV-2, one of 44 coronavirus species known at the time? And so, as Eric mentioned, I was an independent researcher, which just meant I was kicked out of my wet lab. And so I started, I took this idea of looking for novel coronaviruses to different hackathons, including the HackSeek Genomics hackathon that was supposed to be happening in Vancouver. It was going to be happening in April of 2020. And so by taking this idea to a bunch of different hackathons, I was able to recruit an international team of collaborators and world-leading bioinformaticians that we really moved the ball forward. And so this wasn't just kind of an academic problem of trying to find new coronaviruses or where SARS-CoV-2 could end up. This was going to have biological impact. So even early on in the pandemic, SARS-CoV-2 was predicted to infect over 80 different species of mammals. And the concern, again, this was, early on that these slides are from, is that if SARS-CoV-2 entered one of these mammals and there is a coronavirus in that mammal reservoir, those two viruses potentially could recombine. And especially if the spike protein is changed in the recombinant virus, a spillback of that virus would erase the herd immunity that we were pushing towards, either vaccine immunity or natural exposure immunity. And essentially, it would restart the SARS-CoV-2 pandemic over and over again. So this was kind of a threat that we saw on the horizon. That was actually quite prophetic. SARS-CoV-2 has now been reported to infect over 24 species of mammal, including stable animal-to-animal transmission in white-tailed deer in Ontario and Michigan, where a alpha variant of SARS-CoV-2 is now essentially in that animal reservoir, the deer, and it will be there in perpetuity, right? So that is now a completely different variant than the one that's normally floating around in people. And essentially, it's now a ticking time bomb to when that thing spills back. But that wasn't exactly kind of the recombinant. It was just it entered an animal reservoir. So it was close, but not exact. And I think that overall, this really now gives us a perfect case study of how a bad coronavirus in China can cause trillions of dollars of economic damage in America just a few months later. And this idea that it's important not just to study known human pathogens or things that are directly infecting humans, but we have to have a better understanding of the viruses in all the animal reservoirs, in all the environments where humans are also present, to understand kind of the whole picture of virus biodiversity. This idea is called One Health, integrated virology research. And that's essentially where we're going towards next. So where do you go looking for coronaviruses if all you have is a laptop? Well, I hypothesized that coronaviruses would have been incidentally sequenced in publicly available sequencing data. And because most people don't look for viruses in their sequencing data, those viruses would have gone missed. And so all we had to do was reanalyze all public sequencing data. Lucky for us, in February of 2020, the NCBI, through a major infrastructure project called STRIDES, decided to mirror the global collection of raw sequencing data, the Sequence Read Archive, onto Amazon Web Services and Google Cloud. To give you some perspective on what that volume of data, like how big that is, currently there's over 60 petabases of sequencing data from a total of over 30 million samples. There's about 10 million RNA-seq samples. So another way to put that into perspective is that if you were to run Illumina machines to generate that volume of data, that represents between 3.7 and $14.9 billion in direct sequencing costs. That doesn't take into account sample acquisition or personnel costs, which you could conservatively multiply that number by 10. So we could say that we are looking at a $50 billion sequencing resource that we're going to reanalyze if we have the computational capacity to do this. And so if you take anything from this project, I really like to emphasize that this Sequence Read Archive, this collection of sequencing data we've been accumulating for over a decade, is really our modern Library of Alexandria for genetics. There are samples from all over the world. There's cell lines here in Toronto. There's sequencing of soil from an ant mound in the Amazon basin, all the way to anal swabs of penguins taken in Antarctica and everything in between. And so where a big kind of classical virome study would be something like 5,000 samples and it would cost a lot of money, we were going to be working on the order of millions of samples in the context of a database that has been growing exponentially for the past 15 years. And so to do this, we had to kind of aggressively cost optimize and efficiently be able to run a sequence aligner on the FASTQ data, essentially, that was in the SRA. And so I won't go too much into the technical details here on how Ceratus was implemented. I have some technical talks on YouTube if you're interested in that. But suffice to say that the key things were that we aggressively cost optimized for the ability to move data, so I/O. The main limiting factor with processing petabytes of data was how quickly could you get that data onto a CPU. And once we solved this by using very small instance classes and kind of fracturing the read data into small packets, we could horizontally scale our workflow very optimally. So we then scaled to about 23,000 vCPUs, which we could run in parallel. So the punchline of all this is that using Ceratus, we can process over 1 million NGS sequencing libraries per day. And the cost of analysis per library is under half a US cent, which essentially means that for the first time, we are able to analyze sequencing data faster and cheaper than the world is producing that data. This is a fundamental shift in the bioinformatic analysis capacity. As global sequencing data is going online, we can sit there and monitor all of it for viruses. And so, when you build a really, really big hammer, all of a sudden you start looking for big nails. And we decided to expand the scope of the project, not just looking for novel coronaviruses, but looking for all RNA viruses full stop. And to do this, we selected one gene, the viral RNA-dependent RNA polymerase, or RDRP. You can think of this as kind of the heart of an RNA virus genome. By definition, every RNA virus genome has to contain an RDRP gene. And we went through GenBank and publicly available data sets at the time. So this is now December of 2020. And we collected and curated every RDRP sequence we can get our hands on. And then we clustered those down to 90% amino acid identity. And that gave us 15,000 known RNA viruses. So that's our baseline. And now to be able to analyze and kind of cluster these RDRP sequences, which could radically change on their exteriors, we focused on a subsequence of the RDRP that's oriented around the catalytic core. So you can see that as the A, B, and C motifs of RDRP. You can see in the conservation track, that's where the maximum information about the identity of the RDRP is held. The way that I like to explain this is that by using this A, B, and C motif, which is about 100 to 150 amino acids, that gives us a molecular barcode where we can very easily do global alignments and compare RDRP sequences to one another in a very tractable way. And so we called this the palm print, and this is operationally the equivalent of 16S rRNA barcodes in bacteria or ITS sequences in fungi. We just invented a computational version of this for RDRP and RNA viruses.
Can you just say a little bit more about what is that? I mean, when you say it's the palm print, is the palm print that full motif, or is it those conserved residues, or is it like a profile HMM? What is it actually?
So it's sequences. So it's amino acid sequences, and the palm print barcode begins at the beginning of this motif A, which we recognize with a position-specific weight matrix, and it ends with motif C, and it includes the variable region. So from the beginning of motif A to the end of motif C, that's about 100 to 150 amino acids. So each one of those centroids at 90% clustering represents kind of like our species-like operational taxonomic unit, or our representative sequence for a cluster of sequences that are like the SOTU sequence.
Okay. But it's basically encoded as a position-specific weight matrix.
No, so the SOTU is encoded as a flat FASTA. We identify them using the position-specific weight matrix, right? So so Robert Edgar wrote a tool called PalmScan, and that's what it scans RDRP sequences and identifies the three motifs to give us the kind of coordinates of where the barcode sequence is.
Got it. Thanks.
Yeah. And so using Ceratus and this collection of full RDRP sequences, we then searched 5.7 million sequencing libraries, or 10.2 petabases of sequencing data. That represents all RNA-seq data and then some. And using this, we discovered over 130,000 novel RNA viruses in a single 11-day analysis. So to kind of reframe that, more dramatically, is it took the global virology community over 130 years to find the first 15,000 RNA viruses, and we found the next 130,000 RNA viruses in under 2 weeks. And importantly, we also included a control of DNA sequencing data in this search. So as you can see, in over 2 petabases of mammalian whole genome and whole exome sequences, we only identified 95 novel SOTU sequences, which gives us an FDR of about 0.0029. Whereas in the remaining 8 petabases, that's where we identified the vast majority of, or all of the 130,000 new kind of viral species-like units. And so if you can now start to think that what before was a computationally very difficult problem, we essentially can turn into a very quick database lookup. So Ceratus, we put all of this data into a free and publicly available web interface. So all the data is in the public domain. It's available on ceratus.io. And essentially the data that's stored there is that for every SRA library, we did this translated amino acid search for RDRP sequences, and then the alignment statistics for the reads for those RDRPs are stored in a SQL database. So what that means is that for every SRA run, you essentially have metrics about how many viral reads were mapped to that library and what was the average alignment identity. So if you have a library and there's 1,000 reads and they're all 100% matched to SARS-CoV-2, you could conclude, well, SARS-CoV-2 is likely in that library. If you have another library where there's a rubella virus match, but the reads are only aligning with 80% amino acid identity, you would say, okay, there's something that's like a rubella virus in this library, but it's probably novel because the alignment identity is below 90%. And so about 60% of the SRA runs that we processed were positive for at least one read. And then we took those aligned reads and we assembled them into what we're calling microcontigs. And then that gives us kind of like an assembly of the RDRP gene. And then we could scan those micro assemblies of just the RDRP to find intact palm print sequences. And that gave us 880,000 palm print positive contigs across the SRA. And so as I mentioned, what was before this big kind of almost intractable problem now is just the database lookup. If you're interested in novel coronaviruses, you just subselect the Coronaviridae family. You take every library here where the average alignment identity is below 90%. So that's what's highlighted. And that's essentially a fast way to show you which libraries in the SRA are likely to contain a novel coronavirus. And so when we did this, and then we went in and assembled those libraries, we found that they were in some very unexpected places. We found 9 new species of coronaviruses, and they were all found in aquatic vertebrates. So we have the axolotl, the seahorse, the fugu fish. And then we went in, assembled those libraries, and started characterizing the genomes that we were able to recover. And we kind of ran into this anomaly where, we were pulling out these contigs and, they had that RDRP that we detected, very good. They had the non-structural genes on those same contigs. But for a clade of these coronaviruses, they were missing their spike or structural genes. And after a little bit of bioinformatics sleuthing, what we identified was that these structural genes were found on a separate contig. So here you can see at the top, each of the rows is from a different library. And in the top there, that's the axolotl. Can you see my mouse? Assuming you can see my mouse.
Yes. So yeah,
no worries. So you can see here, each of these are different libraries. So for these axolotl coronavirus, these had over 40,000-fold coverage in some of these libraries. And we were getting pretty much nucleotide resolution endpoints. So this, plus a little bit of molecular data that I got from a kind of friend at UBC, we were able to conclude that this clade of coronaviruses are actually segmented. So the structural genes occur on one contig or one segment, and the non-structural genes occur on a second segment. And then that violates the textbook definition of coronaviruses, because if you open up your textbook, they're always supposed to be on one molecule. But here we have very good evidence that we have segmented coronaviruses. And whereas if you made this observation and you only had one virus, it would be really difficult to make that conclusion. But when you have 7 coronaviruses monophyletically with pretty much the same nucleotide endpoint, we can pretty robustly say, yeah, there's something weird going on, like segmentation with these coronaviruses. So that's not the only thing that we could do. We can also kind of frame the problem in a different way, not just where are the novel coronaviruses, but what about coronaviruses that we know of the species already, but they're showing up in a place that they're not supposed to, specifically in a host that they're not supposed to. So we call this virus host discordant libraries. And that type of discordance is fairly rare, but in one case, we identified 2 immunocompromised children who had a fever of unknown origin. And when we went into the sequencing data, those 2 samples had high levels of murine hepatitis virus, which is another beta coronavirus, as the name suggests, that has been known to infect mouse livers. And here we see it in child blood. And so this is now where we kind of get to that limit of like, what can you conclude bioinformatically? So we can't actually say that this was an infection of MHV in these children, although that is a plausible hypothesis, because some of the other coronaviruses that had this virus host discordance were, let's say, bat, mouse, or pig coronavirus, but we were finding them in plant transcriptomes. And so, it's really important to conclude that the presence of nucleic acids within a sample is not really proof that an active infection is occurring. But we can actually view this data in a different way that is meaningful. So I like to use this example of the bat coronavirus that was found in a corn transcriptome. You would say, okay, well, that's some kind of contamination. That's not really important. Well, yes, that's true. But if we kind of expand our mind about how we think about the data, it actually holds some very important information. The bat coronavirus was originally identified in Hong Kong in the 2010s. So I think it was in the early 2000s. And so we have this bat Hong Kong virus. The corn transcriptome that we saw, that was in mainland China in 2015. So all of a sudden, this bat that is previously epidemiologically linked to Hong Kong and an earlier time point, we have now made an observation of that same virus or something that's very close to it at a later time point in a geographically different area. And so what we think happened was that a bat flew over this cornfield, pooped on the corn sample, and then that's what got picked up in the RNA sequencing. It was purely incidental. It wasn't very high coverage, but that now gives us a geospatial and temporal information about that bat coronavirus that we otherwise wouldn't have. Obviously, if this was something like SARS-CoV-2 and we picked it up in a corn sample in 2015, that would be, that's essentially what we were looking for with Ceratus originally, is these kind of incidental observations of a virus where it wasn't supposed to be. And so another application of Ceratus has been that because we release this data freely and publicly, other groups have started looking at different types of viruses across the SRA with it. And so using an earlier version of Ceratus that contained some DNA viruses, this group led by Caleb Loro at Stanford, he identified that CAR T cells, so this is an FDA-approved therapy where you take T cells from a cancer patient, you engineer them in the lab, you culture them for 20 days, and you reintroduce them into the patient. What he observed was that anytime there were T cells that were taken from a patient and they were cultured for a long period of time, like in the CAR T cell therapy protocol, that led to a low level, kind of sporadic reactivation of this virus, human herpes virus 6. And then that virus could then propagate in vitro. And, most of those were just experiments. It was someone doing a 30-day culture of T cells. It didn't really mean anything. But an important kind of link, and where this is now kind of getting interesting, is that if that happens in CAR T cell therapy, which they showed that it does, that you're now giving an immunocompromised patient a reactivated virus, and human herpes virus 6 in this case. And previously, HHV6 was linked to causing one of the worst side effects of CAR T cells, encephalitis. And this is a really devastating encephalitis for the patients. So where people thought normally that the virus was latent in the person, in the patient, and something in the CAR T cell therapy was reactivating the virus in the patient, here there's evidence that actually it's the CAR T cell therapy, like the cell product itself, where the virus reactivation is likely to be occurring, which essentially means that a simple qPCR test is a way to stop a devastating side effect of a FDA-approved therapy. And which now kind of sets the context I think, globally of where this type of virus surveillance is going. So the World Health Organization put forward this really beautiful white paper on global genomic surveillance strategies. And one of the key things that they emphasize is that genomic surveillance, as we saw with SARS-CoV-2, is going to become a bigger and bigger part of public health and public health decision making. There still is a very large technical and financial cost of doing genomic surveillance, especially in resource limited settings. And this is emphasized that it's not just generating the data, it's also in the analysis and interpretation of that data. How do we make this data useful for people to have meaningful kind of results or do something? And so I view the objective of my lab now in Toronto as building these tools so that we can deploy them freely and broadly and empower our colleagues in resource scarce settings to make the most use of their genomic surveillance data. Which kind of leads to this big, I think, inequality in data that we're all facing. The amount of data is growing exponentially. It's enormous how much petabytes of data is available to us. And one of the first major problems that we have to overcome is taking that data and being able to create human interpretable knowledge from that data. So I view this as like a publication or a Wikipedia page or a figure that a human can read, because by building up the base of human knowledge, that will then inform us to make wise decisions. So like when the next pandemic rolls around, when is it wise to close down airports? When is it wise to enact different public health measures, right? So that little bit of information is pretty much little binary decisions are the pointy end of this. And so I view, the objective of computational virology now to really live at this interface between enormous volumes of data and creating meaningful knowledge to then power the wisdom of the field of virology. And I think, as I alluded to before, all of our data is from public domain data, and so we also release it into the public domain immediately. And all of it is available on Stratus.io. We spent quite a bit of time building user interfaces so that, again, we can build that knowledge layer so that we can then empower that wisdom layer. So one example is an interface called PalmID. And here, simply what you can do is you can take any RDRP sequence, known or novel, like I will show you with a rubella virus example. And then what it will do is it will look for that RDRP sequence, align it to our database of SOTU sequences called PalmDB. And then depending on where each SOTU was observed in the sequence read archive, pull out and aggregate the metadata for the virus of interest. And so what that actually looks like with the rubella virus example is that 60% of the SRA data, we have geographic data. So you can see here, rubella shows up in some human eye and skin metagenomes in North America. That's expected. But we also make an observation that there's an 80% amino acid identity rubella-like virus in a bat metagenome in China, again, sampled in 2015. So here we now can extend the geographic range of rubella-like viruses. And this all takes about 2 minutes and is essentially free because it's all available in the web interface. It costs about 1 or 2 cents per API query. And so using PalmID, what used to be a very long and tedious process, essentially is available in the web browser. You put in your virus of interest, you know exactly which libraries to go look at, and then you can go in and assemble as much of the genome as possible. So we identified there was known rubella virus and 2 other ruby viruses, Rahugu and Rastrella. We identified Ruche ruby virus in a horseshoe bat, and then 2 other species of ruby-like viruses, one in salamander and one in frog, which we call Rumple and Ruffle, respectively. And so now you can really start to reimagine that kind of sequence read archive as this passive planetary-wide viral surveillance network. As the amount of sequencing data has been growing exponentially, essentially since the completion of the Human Genome Project, doubling every 18 to 24 months, that trend is expected to continue through the end of the decade. And so as billions of dollars are going to be invested in generating sequencing data for a trivial cost of something like $80,000 a year, we can essentially turn this into a passive kind of biosecurity blanket for all of us. We can monitor the data and observe when is a dangerous agent going to occur. Or more importantly, building a database that we can then look up quickly so that we can interpret viruses when they do show up in places that they're not supposed to. A very good kind of example of this is if a child shows up with a fever of unknown origin here at Sick Kids Hospital in Toronto, it should take 30 seconds and cost us 1 cent to be able to link that virus in that patient's blood to a camel sampled in sub-Saharan Africa in 2012. And essentially, that's the system that we're building today. And the beauty of seeing something and building something is not just kind of doing it ourselves, but seeing in all the creative ways that other researchers are deploying it. So I have no background in plant virology, but it's been really great to see some plant virologists take and use Serratus to extend the kind of geographic range of the viruses that they were interested in. In this case, I believe is a tomato plant, Ilar virus, right? And so, that wasn't my wheelhouse at all, but by making this data available and very easily to query, you can then empower virologists to extend the geographic range. So we can now monitor, this is a great example of monitoring agricultural pathogens across the world, because all that agricultural data has been generated, and essentially now we can just go back and look using a different intersection. So if the data is growing exponentially, does that mean that we published this work a year ago or over a year ago now? So should we have found another 150,000 RNA viruses by now? Well, no. That's actually probably a vast underestimate of what's out there. And that's because of a technical limit in the Serratus workflow. So we use this translated amino acid search called Diamond, which is essentially like running BlastX on short reads. And as you can see here in some simulated data, as the percentage identity of a virus pair, so of RDRP and another RDRP pair, as the percentage identity between them drops, the probability of recovering the reads from the unknown virus also begins to decay. And so by the time you get to 50% amino acid identity, you have less than 50% probability of recovering the RDRP of that known virus. And when you look at the global, this is a histogram now of each of the barcodes that we recovered in that first paper, you can see that it really grows around 60, and then it begins to drop off. And that drop off is purely technical. That's because the way that we did the search was going to be missing a lot of the viruses. So we've only, this 130,000 that we found in that first pass is the tip of an enormous iceberg. And we have pretty much no indication of how big that iceberg is or how deep it goes. I like to say that essentially what we've done is skim the surface of a giant ocean from the beach where we could reach the ocean, right? And then there's all this vastness out there. And so one way to address this problem is kind of the classic SciBlast method, right? You just take it and you build, like for every new virus that you find, you repeat the search and you build it out in n steps. Well, that's a pretty tedious job. And, I was always taught that a good scientist is a lazy scientist. And so we thought, well, what if we spend a little bit extra effort, try to find a bunch of highly diverged RDRP sequences, and then use those for the next pass of Serratus. And that's how we're going to explore deep viral space. And that's essentially what we did with these dark viruses, or ones that we couldn't hit in the first pass of Serratus. So we took all 50,000 sequence assemblies that we generated as part of the first pass of Serratus. We clustered all the open reading frames in about 39 million. And then with no semblance of elegance, we just threw collab-fold and HH-pred at these 39 million clusters, predicting de novo, using collab-fold, just predicting what these structures that were being output. Because what we know is that structure alignment can go much deeper in evolutionary time than sequence alignment. And so then we have all these kind of spaghettis. By clustering the spaghettis, we could then say, okay, we know that this is what the RDRP catalytic core looks like. Which which structures that were predicted are able to have something that looks like the catalytic core of an RDRP as well. And so just a comparison of how the relative performance, this is an example of RDRP viruses that are like very, very diverse from one another. And you can see that it's kind of at the fringes of what MMSeq is able to identify. But when you use fold-seq, again, doing this structure-structure comparison, you get far more sensitivity to go and compare, like, say, a negative stranded RNA virus, and then be able to identify a positive stranded RNA virus using that structure. And so that's essentially what we did, is then we took those predicted RDRP sequences, and then we reprocessed now 7.5 million RNA sequencing libraries. And so you can see in 2022, where we found that 130,000 RNA viruses, that was, our nice paper. These are kind of some of the other major virome studies that have come out since. And then with this version 2 of Ceratus, we then identified another over 250,000 new RNA viruses, putting the total count to just over 500,000 RNA viruses today. And so this is why I like to say we are now just entering a platinum age of virus discovery. We are seeing viruses that are incredibly diverse from all facets of biology. And essentially, whatever creative question that you can come up with, that's essentially the kind of million-dollar question that we should be going after. Can we find new viruses associated with human cancers? Which diseases of unknown etiology are caused by a virus? Can we find new RNA viral vectors that we can use across the domain of life? Or can we identify the next major pandemic agent, the next big one? So I'll just walk you through kind of one example of how we're using this data now is, Alzheimer's disease is a disease of unknown etiology. People don't know what causes it. And we can come up with kind of a very, wishful thinking hypothesis saying, I think that a highly diverged RNA virus may be one of the causes of Alzheimer's disease. And so essentially what this is saying, can we find new neuroinflammatory viruses? And so the way that we did this is that we took 1.6 million human data sets that were processed in Ceratus. We processed their metadata to identify any tissue metadata terms that were present in those libraries. And what this has now created is kind of like a digital human virome generated from 1.6 million samples. We can then laser focus on the 85,000 human central nervous system data sets and use that as a subquery and say, okay, which highly divergent virus occurs in that 85,000 human central nervous system data? And that is, the top, pretty much the first hit that we looked at was this bioproject or the KNOW study from 2017. And so what they did here is that they took 3 human cell lines, neuronal stem cells, neuronal differentiated cells, or monocytes, and they infected them with 9 different strains of the parasite Toxoplasma gondii. So this is a eukaryotic unicellular parasite that goes inside human or animal cells. And what we found was that, we identified the 2 neuronal cell lines, and we found that the virus was in only in 2 samples, and those 2 samples were both annotated as belonging to the same strain of Toxoplasma gondii, this red one called RUB, R-U-B. And then that gave us kind of a natural experiment. Well, we know that we found it in 2 out of 2 and not in any of the other controls or the other toxo strains. But we didn't look at the macrophages because that was a part of our original screen. And so we hypothesized, well, if this virus is associated with a specific strain of toxo, then we should also see it in the macrophages. And that's exactly what we found, is that this highly diverged virus also co-segregated with the macrophages, leading us to conclude that this virus is associated with one particular strain of toxo. And so we called this virus apocryptovirus, Odysseus. So it's named after kind of a Trojan horse, because as you can see, the AO virus, its expression was really strongly linked to this atypical RUB strain. And we didn't see the virus, any reads to the virus and any corresponding DNA sequencing data. So this is an RNA-specific element. And so then we can ask kind of a more general question. Okay, we're looking at human neuronal data. Where else do we see this AO virus? And so we requeried all the Ceratus data. So that's 7.5 million sequencing libraries, or almost 200,000 bioprojects. And there was one other project in the SRA that had this virus in it. And lo and behold, that was also a study of Toxoplasma gondii, but now it was done in murine PBMCs, so peripheral blood monocytes, and exactly the same strain of toxo, the Rube strain, was the one where we found the virus. And in this study, they had 29 other strains of toxo. And one other strain, cougar, was also positive for the virus. So we now have 2 strains of Toxoplasma gondii that are infected by 2 different strains of an Apocryptos odysseus virus. And then you can say, okay, well, are the Rube and the cougar Toxoplasma gondii related to one another? And this is actually just kind of the same thing. That's not true. So we built a phylogeny using the transcriptomes from the toxo, and the Rube and the cougar are as divergent from one another as toxo could be. This is about 1 million years of T. gondii evolution. But that virus that's linking them is really strong indicator that this is something that was horizontally jumping between different toxo strains, like a virus would. Now, as we were learning about the Toxoplasma gondii, because I didn't know too much about it, so this is a parasitic infection. It infects billions of people across the world. Something like 60% of the world's population is going to be infected by toxo at some point in their life. And it can form these lifelong cysts. And the vast majority of the time, toxo is completely asymptomatic. People don't even know that they have it, or they have very mild symptoms. But sporadically, toxo, you can get very severe toxoplasmosis. So incidentally, the Rube strain, which was isolated from French Guinea, that was isolated from an immunocompetent soldier who had multiple organ failure and very severe disease. Okay, the cougar strain, that was associated with a 1994 toxoplasmosis outbreak in Victoria, BC, where a mountain lion pooped into the Victoria watershed and over 7,000 people were infected. That outbreak was recognized because the cougar strain is another example of a hypervirulent strain of toxoplasmosis. Okay, so now we have a very interesting observation. 2 very distinct strains of toxoplasmosis, but they are both hypervirulent. They're both very good at causing disease in humans. And so right away, I was like, oh, well, maybe the virus is causing the disease. And as we went and read the Milo 2013 study, that's where this the cougar strain was found in. Later on in the paper, they actually have this paragraph where they specifically say that only a few atypical toxoplasma strains are capable of inducing high levels of interferon expression, the most potent being Rube and cougar. So, if you remember back to your immunology classes, interferon is very often produced in response to an infection. Interferon beta, as they showed in this paper, was being produced in response to a nucleic acid. And so, they did this genetic knockout where they knocked out TLR7 and IRF3. And so what they showed was that this interferon expression that is linked to the hypervirulence of Toxoplasma gondii was being caused by nucleic acids, specifically RNA, that are in those Toxoplasma strains. So this study, they didn't know about the virus being present in their toxo, but when we saw this, we're like, well, it's the virus in the parasite that's going to be driving it. And so as a minor observation, when they were doing gene set enrichment analysis, this is differential gene expression analysis, they did note that there was an antiviral response that was associated with Rube and cougar. And so we decided, okay, can we independently test that hypothesis? What if we go back to the No 2017 study where we first found Rube in human neurons and ask, is there an antiviral response in that data set? And that's essentially what we did. We went back to that first study, we did differential gene expression and GSCA, and lo and behold, the Rube strain, while there are other strains that cause inflammation in general, the Rube strain is the only one that causes a specific cellular response to viruses, right? And again, that Rube strain also induced interferon beta. And so, this is the model from the 2013 paper where they said, okay, the parasite gets inside the cell, and then these exotic strains, something's going on there. the strains, the toxoplasma is not as fit. It doesn't form cysts as readily, but something about the parasite's DNA or RNA is triggering this immune response that's driving interferon beta. And we now have a simplified model where we're saying, well, actually, it's the viruses that are in the parasite. You get Toxoplasma gondii infection. If that parasite is infected with a virus, that virus is then secreted inside the host, the mammalian host cell, and that's what's going to be driving this type 1 interferon response. So in effect, the virus of the parasite is the thing that's causing the disease in human cells because it's just incidentally there interacting with the cellular, interior cellular environment of the mammal cells. And so you could say, okay, well, is this some kind of one-off virus? It's a fairly strange type of virus called the narnavirus. Well, we then decided to ask a broader question, like what other viruses are in the phylogenetic neighborhood of this Odysseus virus? And we were able to identify 18 additional new species of virus across 195 samples. And what was very distinct about all of these samples where we were finding these apocryptoviruses is that they were found in vertebrates or an AP-complexa data set. And then we could say, okay, the no study was annotated as being human, but it had a parasite in it, an AP-complexa parasite in it. So what if we were to generalize that kind of test and say, okay, everywhere where we see this RNA virus, is that predictive of a parasite being present in that sample as well? And so we essentially formulated this as a Fisher's exact test where we could take, say, chicken samples, because one of them was in this Eimeria parasite, one of these apocryptoviruses. So we have observed in the SRA over 22,000 chicken samples, and under 1% of those chickens are positive for this parasite. If we subset and then say, okay, what about the chickens that are positive for the virus? Well, all 8 of those chicken samples are also positive for the parasite. And we repeat this for all the species, for all the different viruses, and every single time we get this very strong enrichment of the presence of a parasite when we observe the virus, which is essentially saying that the virus is predictive of parasite co-occurrence in those samples. Again, strengthening that link that these viruses are likely infecting the parasites and not a direct infection of the mammalian or vertebrate hosts. And so this is kind of an example of that, like, again, what do you do with this large swaths of data? You do these large unbiased screens. We had this very specific question of, like, can we find a new neuroinflammatory virus that, is a cause of Alzheimer's? Do I think that this is an Alzheimer virus? Probably not. It's a fairly rare virus. Alzheimer's is fairly common. But this definitely is a very clean mechanism for a virus being a neuroinflammatory agent, where it can use the parasite to hitch a ride and drive neuroinflammation. So this is unexpected, but that's what I'm starting to expect is as we go into large unbiased screens, we have to expect the unexpected. And so kind of one of the big conclusions I like to finish is to have a bit of perspective on all of this work, Serratus and this type of virus discovery. It's not really about taking the known virome and we expanded it tenfold, we can pat ourselves on the back. I think this is a very short-sighted view of where the field is moving, because where I think the perspective that you have to take is that all of this expansion that we did is still a tiny fraction of the estimated trillion viral species that make up Earth's virome. We are at the very beginning of a new era of virology, of actually being able to characterize a significant fraction of the biodiversity of viruses that are out there, and trying to understand entirely new facets of biology, including molecular virology. That's where I think the kind of excitement, that's where I think the future of this is that the next few years are really going to change the game. Because if you combine this exponential growth in data production and increases capacity for data analysis, we're on track to characterizing over 100 million RNA viruses by the end of the decade. And I really view that as kind of the main objective of the lab, is I want to be the one to hit that 100 million RNA virus mark by 2030. And so it's not really just about getting to that number. It's about what new and interesting ways can we use that 100 million RNA viruses to make meaningful discoveries. Which one of those 100 million is the one that's going to cause Alzheimer's or irritable bowel disease? Or which one is going to be the next pandemic agent? That's the more important question to ask. So this, as I mentioned, is the basis of our lab in Toronto. It's the Laboratory for RNA-based Life Forms. I am recruiting. If you're a graduate student or a motivated postdoc, shoot me a CV and a cover letter. Our website is rnalab.ca. So I, again, would like to think that this work wasn't just done by me in isolation. The Toxa work was this group effort from the undergrads and engineers in the lab over the summer, but really the Stratus project and then the kind of other consequences of that project really was this group effort from that hackathon. And to recognize that group effort, when we published our paper, finally, all 15 authors were listed as co-first authors. And I think that was kind of like the minimum that I could do, because those were the people who were willing to take a risk and volunteer on kind of this crazy project when I was like pitching it and trying to get everyone on board. So thanks to all of them. This really, none of this would have happened without that team in place. And with that, I'm happy to take any questions and see where we take it from there.
Thank you very much, Artem. That was just an amazing talk. We welcome any questions from the audience. Just post them into the YouTube live. I've got a couple questions of my own. So you've mentioned a couple times finding dangerous viruses, and I mean, I don't know exactly how that works. You obviously have a lot of viruses. How do you find the dangerous ones?
I mean, that's kind of like the million-dollar question, right? Is how do you prioritize, right? So what I like to do is like essentially every time we do one of these like case studies like Toxo, it's you go through the work, right? You do these analysis, and then you have to kind of like reflect about, okay, like why did I do that analysis? What information did I use to link this like narnavirus? And narnaviruses, one piece of context. They're like, if you look online, they're all fungal infecting, or they're known to be fungal infecting. So people think of them as like plant pathogens or fungal pathogens. And so if that shows up in a human brain, 99.999% of virologists throw that out. They're, oh, that's contamination, right? But for us, we were saying, no, like let's actually dig at that a little bit more. And in that case, the key was that was associated with an interferon response in the same cells, right? So the virus correlated to interferon response in that case. So one thing that we can do is use additional layers of information like host gene response to be able to prioritize, because then you can say, not only are we observing this virus, but that virus is now also correlating with a biological effect on the system that we're interested in that case, humans, right? So like that would be an additional layer of like priority. Obviously, phylogenetics is probably the single biggest factor. If I see a new beta coronavirus, kind of doesn't matter. Anything else doesn't matter about host gene response. Like that's a priority. So just knowing which viruses do cause disease, and then saying things in the phylogenetic proximity to known disease-causing viruses, that's also going to be of interest. So that's actually why we chose the rubella virus example. So back in 2021 or 2021, I think. So rubella virus was the only known virus in that kind of family for the longest period of time. Then there was a capybara and a donkey in a German zoo that had encephalitis that was fatal. And they eventually tracked down that like, oh, there's this like tiny little signature of a rubella-like virus. But think about that, a donkey and a capybara. Capybara are those big rodents, right? Those are very different animals. So essentially what they were saying is like, this rubella-like virus has a high zoonotic potential because it's made this big host jump. They eventually found it in a rodent reservoir. So the mice were running between the zoo cages and then giving these animals encephalitis. But that information that something is proximal to rubella virus, which we don't like really worry about, but the next proximal thing is actually a highly zoonotic viruses. That changes the classification of all the rubiviruses, where now all of them have this like zoonotic potential. It's a very kind of messy system because how do you measure zoonotic potential? There's not really a good metric for this. But if we're able to like say take that as another quantification, then that is another thing that can help us kind of make this decision of this is a priority, right? And then the last one would be like where is it occurring? Is it showing up in a soil metagenome? Probably don't care too much. Is it showing up in a kid's brain? Probably going to care if it's there, right? So by using different layers of metadata, I think that's another thing that like you can kind of fold in and help with the prioritization.
That's cool. Before we move on, your Trojan horse virus. So the reason it was making people sick is because it was just like making a big interferon response, and that was causing.
That's the hypothesis. So those strains of toxo do cause like a very strong inflammatory response, right? And the virus is the most likely agent that's causing that inflammatory response. We don't have that molecular data to be like, yes, that virus is going to make people sick. But this is where a few emails and some handshakes, and hopefully we'll get some of that interesting data. It'd be really great to like take the virus, put it into like a regular like non-pathogenic toxo, and see does it gain interferon and hypervirulence, or if you take the rube and the cougar and you cure it of the virus, then does it lose that inflammatory potential, right? Because then we can actually get to molecular necessity and sufficiency and obviously rescue, which is like, that's the bread and butter. And so I view computational virology as like, we're not going to do that work. We can't make that conclusion, but I can tell you exactly which samples to look at and exactly what the virus, you know what I mean? To be able to pick that out of 7 million and say, this is worth your attention. That's my job. And that's like the game that we're playing, is how do we distill down enormous volumes of data into these very specific molecular hypotheses that can be tested and validated quickly in the lab.
And I'm sure you're already thinking about this, but it seems like, is there some way that the first cut of that could be automated? Right? Like
everything can be automated. That's like, like my job is to make myself obsolete. Yeah. And so like I think I kind of mentioned like everything that we do, every analysis that we do, all the phylogenetics, like it's really about like atomizing it, making it automatable. And then what I eventually envision Ceratus and like its derivatives to be is like, imagine an encyclopedia that will write a Wikipedia page about every virus, including viruses it's never observed, by pulling data from the related viruses, right? Like a self-writing viral encyclopedia. That's the end goal. Because we cannot do this for 100 million RNA viruses. That's not tractable. It has to be done computationally. And so building that infrastructure, that's the goal.
Awesome. Can we talk a little bit about RDRP and the role it plays? I mean, obviously it's central, and if you're going to have an RNA virus, you need it. Are there other classes of enzymes that do that same function? Or, I mean, I don't know, is that a limitation, or do you feel like if we can classify everything that has RDRP, then we're good?
I mean, like there's always holes, right? Like it's biology. It wouldn't be biology if there weren't exceptions. So there are, I mean, obviously, well, DNA viruses, retroviruses, right? Like that's not going to work there. You could do the same kind of Ceratus thing for those type of viruses. You'd have to use reverse transcriptase or different set of marker genes. The RDRP at low levels can integrate into genomes, and then you get these like degraded RDRP sequences, kind of like lines, like if they co-opt or if a line reverse transcriptase inappropriately integrates it into the germline. But that's a very rare event. So like, it's well under 1% of the RDRP sequences are these endogenous viral elements, but that's a problem. Yeah, I mean, there's also molecular problems with RDRP. Sometimes there's been a discovery that like some RDRPs are split into two halves, where motifs A and B are one protein, and motif C and the rest of them are in another one, and then they come together. So it's like, imagine your hand is like cut in half, and the two proteins have to come together. The Pomperin system doesn't work for that, right? It can't accommodate it yet. So that's another limitation. But I think that, like, for me, RDRP and RNA viruses, that's the proving ground, right? Like, it's a well-behaved problem, and it's sufficiently difficult that, and obviously it's diverse and rapidly evolving enough that if we can solve the technical issues with RDRP, we know how to solve them for other domains of viruses or other kingdoms, right? So this is like, all we have to do is get this thing right once, and then we can generalize, I think, in a meaningful way. But so that's why I've been like laser focused on RDRP, is I just don't have the bandwidth to do everything all at once.
Yeah. I feel like I like to keep on asking more questions, but I do want to end on like, do you have any phylogenetic challenges with what you are doing so far? Like,
yeah, so it's impossible to build a phylogeny for all RDRP. That's like one of the big ones is, and this is like, it leads to a lot of kind of like hand wavy classifications. So that, I mean, that's a kind of big one. One of the main things with phylogenetics is, I think, not just phylogenetics, but phylodynamics, right? Like, how can we use this evolutionary relationship information? Like think about what I told you about the rubella virus example, or the Narna virus, how we use these like phylogenetic relationships and their associated metadata to then infer data back onto like the thing that we're interested in, right? So that's why I kind of had these case studies, and that tree is always the rooting for us. But it's not about just building the phylogeny. It's like we almost need to build phylogeny and have better state or feature descriptions on the space of a phylogenetic, like on an evolving protein sequence, right? So like, how do we take the space of a RDRP evolving, and onto that map on the potential to infect humans, say, right? Like, what's the decay look like of that? How do we do that type of modeling? That's really where I'm kind of grappling. I don't have good answers, and that's, otherwise I'd be showing you a bunch more data on this kind of thing. But if you're talking about like, there's a huge intersection, obviously. There's an amazing application. It's just, I don't think that the language and phylogenetics, at least my understanding of it, isn't mature enough to be able to kind of do that level of modeling that it really needs. But if you're listening and you think you can do it, send me an email.
But I just to ask a little bit more, I mean, what you're talking about is surely having to do with other enzymes rather than RDRP. And so when you say mapping these like phenotypic characteristics onto RDRP, like RDRP is like the train track, and then the interesting stuff is happening on the cargo that's riding along the train track, it seems to me.
Well, maybe, but that's something that you can model, right? That's like kind of my entire point, is that the RDRP it's like a unit of measurement in this very complex space. And obviously, like, you're right, other proteins, right? Like, does it have proofreading activity? Does it have a methyltransferase? That's going to confer a phenotype on the virus and like its properties. But say like the viruses that are human infecting are not monophyletic, right? Like flavivirus. So like, the things that there's like, you go down this coronavirus tree and you get to like axolotls and seahorses, and then eventually you can pop up in like the mesenos or the arteries, and then get back to human infecting potential, right? So you can't say that like, oh, everything around it is what dictates it. It does dictate the decay of the trait locally, but it doesn't dictate like the global property of like, oh, that's the thing that is like human infecting. Sorry, my only point to more like clear answer is that, can we use the RDRP as like a metric to measure, but we're proxying the evolution of the other proteins, right? Because they should, like, as long as you're kind of assuming that the relative clock rate of the proteins is comparable, right? Mm-hmm. As long as it's like more or less a linear relationship between RDRP rate of evolution and like say methyltransferase rate of evolution, then you should still be able to make inferences from the RDRP phylogeny.
Got it. Okay, well, thank you very much. That was, again, a really beautiful talk, and looking forward to the rest of the series. All right, thank you, Tim.
Thank you very much. Take care.